Well, South Africa has taken a major step in the fight against HIV, with the first group of HIV-negative volunteers enrolled in what could be a groundbreaking vaccine trial. The brilliant 011 study, launched at the Desmond Tutu HIV Foundation site at the Groote Schuur Hospital in Cape Town, could mark a turning point in global efforts to end the spread of HIV. One of the people involved with this project is Professor Glenda Gray, distinguished professor and leading health researcher with Wits University. Good to have you on the program this evening, Professor. First things first, let's just set the scene here and talk about the point or the purpose of this trial, what it will entail, and what we hope to achieve at the end of it all.
So yes, this is a first in human, first in Africa study, and it's what we call an HIV discovery program, where we try and where we are trying to induce neutralizing antibodies in people who we vaccinate. So this is a very special combination at a special target of the HIV envelope with an adjuvant that's designed to augment the immune response. And so what we do hope is that we're able to teach the immune system to induce these body neutralizing antibodies so that we can teach the immune system and understand whether we need to make a next generation vaccine or whether the vaccine that we have is sufficient and how we can augment this. So this is the first study. It's 20 participants. If we find that the vaccine has a good immune response, we will then proceed to larger studies and hopefully augment the vaccination regimen so that we can find a vaccine that will induce the neutralizing antibodies that we're trying to find.
As ever with this sort of study, Prof, the question is, how much closer does it bring us ultimately to a cure for HIV?
Well, the approach that we're using is both therapeutic and preventative. So we're hoping that these vaccines could be used in people who are living with HIV as a way to improve outcomes, to make a functional cure, or to help with people who have struggled to have their medications. So we see both into both applications, and we'll start off with HIV prevention, and we'll see then how this vaccine behaves with people living with HIV, where we'll try and see if we can induce the immune responses that we're looking for.
So we're starting with this batch of, I think it's 20, right, HIV-negative people. At what point do we move on to people who are living with HIV? And what might the differences be in terms of the efficacy of the vaccine at that level?
So we're trying to do two things, but in both people living with HIV and people not living with HIV, we're trying to induce these body neutralizing antibodies that will contain and neutralize the virus. And so once we see that this vaccine works, so we want both prevention, that's very important for us, because we do need to stop the HIV acquisition. But at the same time, if we can contribute to a cure, it'll help us alleviate the long-term management of people who have to use ARVs for the rest of their lives. So it's a dual process, and I think we must make sure that we pursue both avenues with this type of regimen.
And on the daily, you spoke to us slightly about this right at the beginning, Prof. What work will be done throughout this process, and what will you be monitoring specifically in the people who are then going to take the shot?
So this is a highly intensive study, and we are taking a lot of blood to try and see how the immune system works. So we do single cell, we do B-cell analytics on single cells, so it's highly lab orientated and focused, so that we can understand exactly what the immune system does when it's exposed to these regimens. And so this kind of work allows us to image our immune response and to, if needed, make better vaccines. So this is a prototype vaccine, and we obviously need to optimize the platform as well as make sure that we can engineer even more apt immunogens to get the immune response we're looking at. So we're teaching the immune system to induce these body neutralizing antibodies, and we believe this is an iterative process, and that we may need to design further immunogens to help us march the immune response to the kind of antibody responses that we need.
And part of the bigger goal is to help with Africa's response to the HIV/AIDS issue, right? As far as an African solution then to the problem is concerned, why that particular focus? Because I know even in the strains that ultimately will form part of the study, it is those that are predominantly found, am I right, in Southern Africa?
That's true. So the vaccine regimens we're using are derived from breakthrough infections both in South Africa, in fact from Soweto, and one in East Africa. And so these immunogens have been designed from people who did get HIV, and we were able to identify special targets from these responses, and then design our vaccines to be tropic to Africa, tropic to South Africa, and to support the East African endeavor.
Just out of interest, Prof, how do the strains commonly found in our region differ from, say, Europe, the Americas, etc.?
So we have, what's circulating in South Africa is a clade C, and this is the largest circulating strain at a global level. It's also found in China and India. In East Africa, we have D, as a clade D, clade A, and some recombinants. And then in the US and in the Americas and in the UK and Europe, we have a clade B pandemic. And often most HIV vaccines have been focused on clade B because I guess that's where the money is. And there's been very little attention to the kind of strains that are occurring in South Africa or in East Africa, and hence the need for African scientists to be involved in the search for an HIV vaccine.
And you mentioned money. Let me ask you then about money, because this time last year we were having very different conversations around HIV/AIDS research and programs as a result of the Trump administration terminating the PEPFAR arrangement. So for this particular study that you are part of, is there enough money in the kitty to see it through its various phases? And if so, who's assisting at this point?
So this was a year delayed. We were supposed to start this trial last year this time. And this trial is now funded by the South African Medical Research Council as well as the Gates Foundation. And we've obviously had to make the trial a lot smaller. In our previous iteration, we were going to be a multi-country study with also East Africa involved. And now we are a single site with 20 participants as compared to a 3-country study with 60 participants. So we've lost time. But we are still eager to make sure that we don't lose the momentum that we now have.
Professor Glenda Gray, always good to talk to you. Thank you very much. Glenda Gray is a distinguished professor and leading health researcher with Wits University here in Johannesburg.